Here is a cardiac myocyte. It is the microscopic cell in the human heart that is responsible for heart contraction. All cardiac myocytes are capable of what we call spontaneous rhythmic depolarization and repolarization. This allows your heart to contract over and over again without your brain telling it to, over 1 billion times in your lifetime. Let's zoom in and see how this amazing cell works. Cardiac myocytes have a cell membrane that separates them from other cells. The cell membranes contain ion channels that allow ions or electrically charged molecules to travel in and out of the cell in a carefully orchestrated manner. Inside the cardiac myocytes are sarcomeres, the protein machinery responsible for contraction and relaxation. Sarcomeres contain myofilaments called actin and myosin. When actin and myosin interact, the sarcomere contracts. When actin and myosin separate, the sarcomere relaxes. How do the myofilaments know when to interact? The ions and their carefully orchestrated movement in and out of the cell tell them. Sodium channels open, increasing the voltage of the cell from -80 millivolts to -20 millivolts. This then signals the calcium channels to open. The calcium ions bind to regulator proteins on actin, which allow actin and myosin to interact and contract. Now, how do the myofilaments know when to separate? ATP, or adenosine triphosphate, which is the energy source for the human body, binds to the myosin heads and disrupts the interaction with actin. This results in sarcomere relaxation. Does this mean that it takes energy to relax, not contract? Yeah, right. Try telling that to cardiologists. Meanwhile, the cardiac myocyte needs to return to its resting electronic state of -80 millivolts. So potassium channels open and resting voltage returns to baseline, and the cycle starts again. Let's look now at what happens to the cardiac myocytes when the aortic valve leaks and causes chronic volume overload in the left ventricle. The heart initially tries to compensate for the extra blood that enters the heart during diastole across a leaky aortic valve, in addition to the normal blood that enters from the left atrium across the mitral valve. It does this predominantly by lengthening the cardiac myocytes and by adding more sarcomeres end to end. Over time, and with more and more volume overload, the heart starts to lose cardiac myocytes or even entire myofibrils. Energy metabolism with ATP becomes dysfunctional. The movement of calcium in and out of the cardiac myocyte and its interaction with the regulatory proteins becomes impaired. Apoptosis, or cell death, can occur, and overall function of the left ventricle becomes impaired.